2021. Here we are, second meeting in a row over Zoom. And boy, it's unfortunate, but it definitely seemed like the best move considering the state of the pandemic that we're still in. So with any luck, we can all move on from this before too long. And certainly as we go through today, we'll talk about venues for future meetings. But I just want to make a couple quick announcements before I hand it over to Sean. The first being that, you know, most of the organization for this meeting falls on the shoulders of the president-elect. So I just really want to acknowledge Sean Marr for his contributions putting this meeting together. It's not a trivial amount of work. So thanks, Sean, in particular. I do also want to remind the students presenting today that there are prizes in at least a couple categories where we have at least two folks giving presentations. So along with that, we talk about a number of other things, including a prize giving at the business meeting, which is the last item for today. So I would encourage everybody, especially in some ways, including the students, to stick around and join us for the business meeting at the end and just kind of hear about a little bit about the society business. And on that same note, for those state representatives who are attending today, we go around the room to kind of discuss the state of mammalogy in your states. So if you haven't thought about it recently, see if you can pull together a list of talking points for mammals or mammal-related science, conservation, etc. And also stick around in particular for our keynote speaker today, Dr. John Scheibe, who graciously is going to give us a keynote address despite the fact that we're remote. So that will be just before lunch, I believe. Sean, I'll hand it over to you. I don't know if you have any other announcements at this point.
No, I don't. I gave everybody who's on the Zoom permission to share their screen. So what we'll do is in about 10 minutes, Nathan's going to kick off the meeting with the first talk. Presenters, what you can do on the bottom of your Zoom window, which I assume most people know what they're doing, you can share your screen and then go through your talk. Hopefully you got an email from me. If you didn't, we got the wrong email address for you. But talk should be about 12 minutes or so that'll leave about 3 minutes or so for questions. Please consider that we also have the transition, the opening and closing of Zoom windows and all that kind of thing. So I might cut it a little bit, like a minute short, which doesn't seem like a lot, but sometimes it feels like forever. But just to kind of keep us on track. When we get to the poster sessions, we have 2 posters. Those will both give quick little brief lightning talks, if you will, to just describe the poster, kind of like a poster pitch, what have you. And then I will open up 2 extra rooms, and then people can just pop in, and the poster presenters can show their poster at that time. Okay? I did not factor in a specific lunch window. I figured people might be sitting at their desks and just eat. So I decided not to do an official lunch break. I can record all the talks if people want me to. I can record them individually and share them. We can make them like just simple links so people can go back and watch them again if they want to. I don't know how people feel about those sorts of things. But if you're a speaker and you want it recorded, let me know just through the chat, and I will hit the record button and send you a link or something like that. Okay? What else we got? A few people, should we talk, we got 5 minutes before we're supposed to talk. Can we talk about a few other things?
Sure, yeah.
So there was, we had some T-shirts that I had planned to order, and then we went to Zoom, so I canceled the order, and they refunded everything. Andrew and I were talking about, and then Lori had asked about T-shirts too, doing some generic T-shirt designs that we wouldn't change them from meeting to meeting. How do people feel about that? And then we could just pass them through. Thumbs up. Okay. Yeah. That'll save the society a bunch of money because we won't end up with leftover T-shirts. We can have a standard design and then change the color every time we kind of run low or something like that. So you should, if you ordered a T-shirt, you should have gotten reimbursed for the T-shirt. All the speakers, student speakers, a few people did paid for the registration. I'm going to refund you the $15. Okay, so you'll get an email for the registration if you're a student speaker. A few people did that, but you don't have to do that if you're the speaker. But some of those people were like, did it yesterday. Okay.
Sean, I got one more announcement just before we get going. So each year we roll over some of the state representative positions. There's 3 that are being renewed this year, which include Arkansas, Kansas, and Iowa. And there's a 4th one. Currently, Nathan Proudman is our student at large, and that comes up for renewal again this year also. So we'll have nominations and elections for those positions, as well as the president-elect in the business meeting this afternoon. So think about that as we go through. If you are a student and you'd like to be involved, I mean, I think we can nominate, you can nominate yourselves or you can get nominated by your advisor or something like that, but also for the state representatives, for the different faculty members. So, yeah, so keep that in mind as we go through today. All right, Sean, are you gonna lead our speakers today?
Sure, I can just introduce folks, I guess. I think we're all here. I think one thing we want to do is just a reminder that if you're here and we're not in a general discussion, please mute your microphone. I think I can mute people individually if it pops up. Also, if you could turn off video during the presentations, sometimes that affects bandwidth for especially students or people at home. I know when I'm at home, I have to turn video off, but that will kind of make it a little bit more seamless for everyone. So, okay. Should we just get started 5 minutes early? Are people comfortable with that? I got 1 thumbs up, a head shake. Sounds good. Okay. All right. So our first speaker this morning is Nathan Proudman, who will be, he's currently our student rep. He'll be talking about non-invasive methods to assess bobcats in Oklahoma. So you can share your screen, Nathan.
Okay. All right. Can everyone see my screen?
Yep, I can see it.
Okay, excellent. Okay, yes, I'm Nathan. I'm a PhD candidate at OSU. I'm going to be discussing one of my chapters of my thesis, which focuses on the spatial ecology of bobcats. So a bit of background, bobcats are widely distributed throughout North America. They've been surprisingly resilient to a lot of the recent anthropogenic impacts over the couple hundred years. However, they're not invulnerable, and there have been examples of local reductions and extirpations as a direct result of anthropogenic factors. But due to their elusive nature, they're often difficult to study and monitor, and there are still things that we are uncertain of in terms of bobcat ecology. And because they are so widely distributed as well, they inhabit a variety of different environments throughout their range. And resultantly, they have different ecologies in these. So what has been shown from other studies is that home range sizes and densities of bobcats in the landscape is largely influenced by prey abundances. But this can also be affected by habitat connectivity, interspecific competition, and also anthropogenic factors as well play into this. Home range selection differs as well between the sexes. Males tend to prioritize incorporating as many female home ranges within their home range, while female home range selection is more motivated by the abundances of prey and availability of denning sites for rearing kittens. And also, these home range sizes fluctuate seasonally, especially in females. Usually they expand during the winter in terms of when prey are more scarce, and then contract during the summer, especially females when they are rearing kittens. And bobcats have multiple requirements to persist. They not only need adequate prey abundance, but also suitable habitat for, as I said before, denning, also loafing and connectivity and other factors that they require. In areas of their range where there is a lot of forest cover, they tend to select for forest edges. They tend to avoid the mature stands and the interior of forests. But in less forested systems, where you don't have this intense vegetation cover, they tend to select for steep, rugged terrain. And so understanding how the ecologies might be different between these more open areas and these densely forested areas is particularly interesting to know, especially in Oklahoma, where we have a variety of different ecoregions. And you see here on this figure our study sites. Packsaddle and Sandy Sanders, they are actually in two different ecoregions of the state, level 3 ecoregions, that is. Unfortunately, I couldn't change the color. It's a very subtle difference. It's copyrighted from the ODWC, but yeah, they're two different ecoregions, and our one in the east is in very different environment. And to sort of just show you the different environments here, our study site in the northwest is characterized by these rolling prairies. And then in the southwest, there's these mesas and juniper, mesquite shrubland, and then in the east, then it's this pine oak forest that dominates. And from studies in similar regions to our study sites, have shown vastly different home range estimates for bobcats. This is by no means an exhaustive list, but these are the ones I could find on Google Scholar. So it does show that there's, even within these short distances, there seems to be quite a disparity in what they predict home ranges would be. So accurately estimating these is, would be very useful. And so the aim of this project was to characterize the differences in spatial ecology between these distinct ecoregions, but using only non-invasive methods. And so to achieve this aim, we have three objectives. The first is to determine and compare relative abundances and densities between these distinct ecoregions, then also calculate home range sizes and habitat preferences or selection, second and third order selection, that is. And then also determine the relatedness between individuals and see whether the home range overlaps differ depending on relatedness and things like that. And so the primary mode of data collection is these hair-snake cubbies. I've mentioned these before in previous talks, so some of you may be familiar. They were developed by Roundsville, but they're basically just corrugated plastic folded into these tent shapes. And we have gun brushes, which capture hair that are placed on the entrances of the cubby. And then we have a carpet square glued on the inside, which we apply scent lure to, and then the bobcat will duck its head in trying to sniff that, and will catch the hairs on these brushes. We increased the number of brushes to include also brushes on the edges of the cubby, because we found that the bobcats were rubbing their cheek on the edges. And we replaced we top up the lure every week, and then we collect the brushes, freeze them, and then replace them with new brushes every week. And so this field season lasts for 6 weeks total. It goes from January to late February, early March. We've had 3 field seasons in 2019, 2020, and 2021. And at any given time, there is about 120 active cubbies, 40 per our 3 study sites. You can see on the figures, the red triangles are locations where we've kept them for all 3 years. But some we've had to move around due to prescribed burns on these study sites. And also as we find just more optimal positions to put them as well. Spacing is more than 500 meters, and they're pretty dense, so we're unlikely to miss a bobcat home range in our. And so once we have our hairs, as I said, they're frozen and replaced. We'll have for each cubby 6 weeks of brushes. Extract the hairs from these brushes, and then extract DNA. And we use, to determine species, we use these fur ND1 genetic primers, which target the ND1 gene of the mitochondrial genome, developed specifically for mesocarnivore hair. And then once we've identified bobcats from these hairs, because we also get possums and other things going into these cubbies, we can then identify individuals. We've not yet begun this, but what we will do is use a suite of microsatellite loci, and then identify individuals based on these allele mismatches. And we can also then determine relatedness between individuals using Graffin's relatedness coefficient or similar, and in some software, probably relatedness. Also, we're fortunate enough to have camera traps, which supply us with some supplemental data. We set them nearby, but importantly with a viewpoint of the cubby, so we can see the animal interacting with the cubby. When we can calculate activity patterns from this, and also identify individuals from palage pattern. And here you see a bobcat in our, using our cubby. And yes, and so we started out with about 60 stealth cam cameras. Some malfunctioned, as some tend to do. But luckily enough, we were given a grant to get more and loan some from a student who was kind enough to lend us some of her cameras too. And so you can see they've shifted around. Some have stayed in the same position, but as we've got more cameras, we're able to put cameras on more sites. And so what we do from these data is construct encounter histories for bobcat individuals, and then model this in a capture-recapture framework, probably using our AICIT approach. We can calculate bobcat densities, and then habitat selection based on use versus availability. And also we can then calculate home range sizes and degree of overlap, and then relatedness and how that factors in as well. We also have supplemental data in the form of small mammal trapping and scat collection. The small mammal trapping we did directly after the cubby deployments until May, and this is at 3 to 4 of our representative habitat types per study site. And we also collected both bobcat and coyote scat throughout the extended field season. And then this will not only provide a lot of the context to why bobcats are making these decisions in terms of habitat, seeing the prey abundances, but also their diet. But also we can get genetic data from the scat too, which will supplement our capture-recapture framework. We're still doing a lot of the genetic work, but we do have the camera trap observations. We're still identifying individuals, but we have we had quite a lot of bobcat observations over the 3 years, 132. But importantly, we see here we had a lot of rabbit and rodent observations on our cameras as well, which is really useful, and also coyotes, their theoretical competitor. We have identified some individuals so far. This is Poppy. She was observed at 4 different occasions at pack saddle. And so we'll be able to hopefully identify quite a lot of individuals from these photos. In terms of activity patterns, you can see here pack saddle and sandy sanders there on the bottom, very similar activity patterns, but James Collins were the bobcats there were a lot more diurnal than what we expected. So there's definitely reason to suspect different spatial and activity patterns. And so the significance of this research, estimating their abundances and densities is of course really useful for management, especially as it differs between these different ecoregions of the state. It'll also help answer a lot of the questions that we have about bobcat spatial ecology, especially the relatedness aspect, I think will be really interesting. And also doing all this non-invasively, if successful, it could open up this method of mesocarnivore research, using only non-invasive methods as a viable method to obtain this data. And just a thanks for the funders, the ODWC and the American Museum of Natural History for that grant. And also thanks to all my field and lab technicians and the WMA managers. And with that, I'll take any questions.
Nathan, I may have missed it. Have you had success with getting mitochondrial genetic sequences out of the hair samples? And
right. So we actually have a lot of hairs. We have a backlog, actually. We've been struggling with different primers. We used 16S for a while, but that one didn't work. But these Fur ND1 primers seem to do the job, but we're just now starting to truck through our huge backlog of them. So they are going, but it's a bit with non-invasive stuff, it's a bit hit and miss, because sometimes we'll have big clumps of hair with follicles on, which will have a lot of DNA, and sometimes it's just a fragment of a hair shaft, and that can be quite hard to amplify. So
Nathan, do you have any evidence that suggests that bobcats are actually marking these sites and that keeps other bobcats from the sites?
Yes, they quite often, the behavior that we observe is that they usually go in, sniff around, and they will urinate and then walk off, or cheek rub and then walk off. So they do definitely try and mark their territory. And then also sometimes it helps with other bobcats will be attracted to the urine as well as the scent there, so they'll go in and sniff that as well.
Cool.
Nathan, I'd like to ask just a methodology question about the hair sampling.
Mm-hmm.
So you collect all this hair off the brushes. Do you assume that all the hair is from a single individual, or do you separate clumps and treat them as possibly it's not a judgmental question, it's a technical question for further understanding.
Yeah. I think we were fairly conservative because I was worried about that. Previous studies have just done all the hairs on one brush were combined, but I didn't want to do that because it could be multiple individuals, even multiple species. So it was, unless it was clumped in a way that looks like that you're fairly certain that it's definitely the same clump wherever it's all come off and it's all together. If it's not like that, then you treat each separate hair as a different sample. And so it was.
So you would end up with thousands of hairs.
Yes, there's a lot of samples, and there is a bit of subjectivity as well in that, but I think we've probably found the right balance, I hope, at least.
Thank you.
All right. What time is it? Let's thank Nathan. Yeah. Let's thank Nathan. And we can shift to Miranda Tiro. Is that, I'm going to mispronounce it. Tirio?
There we go. That's okay.
Okay. She's our next speaker. She's a PhD student at University of Oklahoma, and she'll talk about mass shrews, Sorix cinereus. So Miranda, you should be able to share your screen.
Can you see that?
Yes.
Okay. There we go. Okay. So, thank you all for having me here today. I'm going to talk to you a bit about my master's work on body size trends in masked shrews. So first I'd like to talk about why I'm interested in body size in the first place. So size scales with pretty much all aspects of an organism's physiology and its role in the ecosystem. So for example, size is related to the size of territory that an individual needs or can defend. It scales with metabolism and thermoregulation, life history traits, and the resources that an individual needs. And so all of this is really important to think about in the context of global change in the Anthropocene. So climate change can affect any one or multiple of those factors that I just listed. And so a change in body size might be a good signal for how a population is responding to climate change overall. And so there's been a lot of interest in these body size trends over the last few years, and so far, it doesn't seem like there are any all-encompassing trends or mechanisms to explain why body size might be changing and how it might be changing. So there's still a lot of gaps in our knowledge as to what drives a particular trend in size in a particular population, in a particular environment. So there have been two major hypotheses that have emerged from the literature on this. So the first, Bergman's rule, is often invoked to explain decreases in size over time, and this is based on the ecogeographical rule of Bergman's rule, which describes a pattern of within a species or genus, individuals living in warmer climates tend to be smaller. And the mechanism behind this is thought to have to do with the relationship between surface area and body volume. So in smaller mammals, like this kind of theoretical mammal on the right here, there's a greater surface area relative to the amount of volume that they have. And so that's more area to exchange heat with the environment. And so as the environment is warming up, it might be advantageous to have a more efficient way of shedding excess heat. So that's one possible explanation, but increases in size are often explained using resource availability. And the rationale behind this is pretty straightforward. As the growing season lengthens and maybe winters are getting shorter and milder, that might give a greater year-round resource availability in some environments. And so that's more energy available to individuals so they can obtain a larger maximum size. But these are not the only two potential explanations. I am interested in demography in particular, because in a lot of animals, including mammals, older adults tend to be larger than smaller adults. And so if those longer growing seasons and milder winters that I mentioned before affect survivorship, right, that might lead to a greater portion of older, larger individuals in a population over time. And if we're not thinking about demography when we're studying these trends, that might look to us like an increase in the average size over time, but really that can be best explained by an underlying shift in population structure. And so I tried to test this hypothesis by reanalyzing and expanding on a study by Yamtov and Yamtov from 2005. And so they studied mass shrews collected in Alaska. And so that's what we're seeing in this picture here. These are these tiny little mammals, one of the smallest mammals in North America. And they found a significant increase in average size over the second half of the 20th century. And they had a pretty large sample size for this. So they used external measurements from 650 specimens that are housed at the University of Alaska Museum. And importantly, to our demography hypothesis, they combined all sexes and ages into one big group. And so the reason that that's interesting to me, particularly because in these shrews, is that they go through a lot of size change throughout their lives. So they actually lose bone and other tissue mass in the winter as adults because the smaller size reduces their energetic demands when resources are very scarce. You can think especially in Alaska, so these shrews are living in places where, you know, above the snow can be negative 40 degrees. And so being smaller means they need less food, and that kind of helps them survive. And so since they're going through this size change throughout their life, that made me wonder, well, does age have something to do with it? Does that affect this trend over time? So I went back to all of those specimens and I determined their age. So I categorized them into 2 age classes. They don't live for very long. So basically we just have young of the year and then older individuals, and then past about 18 months, they tend to not survive. So the younger age class is what I call juveniles for this study. They were collected in the same year as they were born in, and they are characterized by relatively sharp cusps on their teeth that still have a lot of that red pigment left. And then the older shrews, which we're calling overwintered because they were collected after they've survived winter. So these are individuals that are at the age where they typically reproduce. And they are characterized by pretty short and blunt tooth cusps with very little of that red pigment remaining. And so for that reason, I actually wanted to title this talk Getting Short in the Tooth, because that's actually what happens to shrews. So to quantify size, Yomtov and Yomtov, they use these standard external measurements that were taken at the time of collection or preparation. So what we're going to focus on is the total length. So just for our purposes, you can just think of this as nose tip to tail tip, and the tail length. I also wanted to add some measurements. So I took some craniodental measurements because I thought perhaps that the increased precision of that sort of measurement might make the signal a little bit stronger or kind of reveal something else about the trends. So what we're looking at here is how I actually measure that in these tiny, fragile little skulls. So I put them on a flatbed scanner in batches of about 10 at a time, like we're seeing here, and took a scan of the ventral sides. That's what we're seeing. And then that let me take a few measurements from that perspective. So I measured a caudal premaxillary length, we can just think of as the length of the skull here, the maxillary tooth row length, and the cranial width. And you don't need to remember all of these long names for the measurements. Just look for a little picture like this to know what measurements we're talking about. All right, so what I found here actually was in agreement with what Yomtov and Yomtov found in 2005. And so in these 2 plots, we've got a year on the x-axis and we've got residual total length on the y-axis. And all that means is that here we're showing the variation in total length or tail length after I accounted for latitude, longitude, and the month the individual was collected in. So this is just the leftover variation after we've dealt with all of those other factors. And so when I combined both age classes, I found significant increases over time in both of those. Excuse me. And then when we account for age, it looks like this. So you'll see that the juveniles, so the lighter green line here, falls almost perfectly on that kind of increasing trend. And then it looks like there's something different going on with the overwintered shrews. So including age in this model improved the model fit. So it seems like, well, maybe age plays some role in explaining these trends. But interestingly, the interaction between year and age wasn't significant in the models, and so it looks like maybe something is going on there, but still kind of a question mark. I found something similar in the craniodental measurements. So with with both of the age classes combined, I found a significant increase over time in skull length, the tooth row length, and then no change in the cranial width. And accounting for age did significantly improve the fit of the model. And even though it kind of just looks to your eye like maybe something different is going on between these two age classes, the age-year interaction wasn't significant in those models either. And so I'd like to talk about, why is that? It seems like maybe something is going on with age, but to me it wasn't quite clear whether there are any significant age-specific trends. And something I'd like to point out is that it turns out that the majority of our specimens in the sample were juveniles that were collected in August. And so that makes sense. That's a time of year that we expect there to be majority juveniles in the population. And so if that is when people are going out to trap them, that's what we're mostly going to get. But something that I've been thinking about is that these longer summers might benefit those growing juveniles, right? So the improved resources in the summer might account for that increase in size. We've also got this reduction in size in the winter that I mentioned earlier. So that's something that's like a completely different thing that's happening with the overwintering adult shrews. And so that makes me wonder if whether juveniles in the summer and overwintering adults in the winter might be facing different pressures on size. And I'm wondering if this could lead to different trends through time. And, there's also the question of, has population age structure changed over time? Excuse me. Sorry, I've got something in my throat today. But to kind of get at these questions, we're going to need a bigger sample of these older shrews. So these are still open questions for me in this project. So to conclude, what I'd like you to remember from this study is that there are no clear trends or mechanisms that explain all of these apparent changes in size throughout time, particularly in the context of climate change. But hopefully I've shown you that thinking about demography and age structure can offer some new insights and explanations about these trends. And this is all important to think about because shifts in size might be a good signal as to how a population or species is adapting to environmental change. So I'd just like to thank everyone for coming today and listening, and I don't know where I'm at on time, but if you have any questions, you can reach out to me in the chat and I'll send you my email, and I'd love to talk. Thanks.
Randa, that was great. I don't know if you know, it doesn't take much to get me excited about shrews. So,
I like them too.
where was your sampling from? Was it from all over Alaska?
Yeah, I don't actually have the map in these slides for some reason, but it was mostly in interior Alaska. Yeah.
Okay. Yeah, I was wondering because, Alaska's a huge place, obviously, and I lived in Anchorage, and it's tropical there compared to Fairbanks in the wintertime. So
yeah,
I just
absolutely.
wondered if there's any kind of latitudinal signal at all amongst.
So in the original study, if I'm remembering, they looked at those they looked at the location as well, and I think that they found that the shrews kind of had an I guess we called anti-Bergmanian. They got smaller at higher latitudes, and I think that we also found that as well. And that might have to do with since their strategy to survive the cold is to be smaller and reduce their energetic demands, might have something to do with that. So where the winter is harsher, they're getting smaller. That's what I would think about.
Yeah. I mean, also the whole like Denell's syndrome, like you were talking about in the winter, they get smaller. I don't know if you know the answer, but it would be cool to know if that varied depending on, where they were too.
Yeah, my hypothesis on this is that in the summer it's getting warmer, longer summer. They have all this time to eat and get big as juveniles, but then the winter is still cold enough that they need to retreat under the snow, and maybe even having more days in the winter that are above freezing might melt the snow and collapse that subnivean zone that they depend on to kind of buffer themselves from the cold. So they might even be exposed to more of the cold in the winter because of climate change. And so my hypothesis is that the kind of difference between their like maximum body size in the summer and their minimum body size in the winter might be increasing. That's what I think. I don't have any data on that right now, but I'd be interested in figuring that out sometime.
Yeah, that's a cool dynamic. Thanks.
Mm-hmm.
Yeah, is there any difference in age class relative to size that you notice or anybody else has been able to notice? I mean,
yeah. Sorry.
So if you get an older adult, more senior, would that animal be larger typically, or?
Yeah. So there were some studies where they were able to. I hope I'm saying this right. Lazaro, I think. They were able to actually capture and release and then recapture some individual shrews. I don't know how they did, because anybody who knows about shrew trapping knows that they'll die for anything. But they were able to kind of measure, like exactly quantify how much mass that they're losing, and they found that I should note that this was a congener of Sork scenario, so maybe something different is going on with these guys, but in this other species, they were able to find that they lose some percent of their mass in the winter, and then they regain it, but like a smaller portion, right? So they don't regain quite as much as they lose, and so because of that, my I think the next logical thing there is that when they're older, when they're overwintered and they're reproductive, they're actually smaller than they were before winter when they were still juveniles. And then we get into the I also found that there was a little bit more overlap between the juvenile overwintered stage than I thought there was going to be, so that might also have something to do with these changes in the environment. Maybe juveniles are able to breed when they're younger, or something like that. So there definitely is a lot of nuance and complication in these age classes that I'm interested in finding out more about.
Well, I think we have to. Sorry, Stephanie. I think we have to move on because it's quarter up. I want to thank Miranda again. And our next talk is by Tommy Herrera, who will discuss a variety of Great Plain mammals and looking at community assembly in different lineages. So take it away.
All right. Everyone hear me? Unmuted?
Yeah, you're good.
All right. Hey guys, so my name is Tommy Herrera. I'm a master's student at Kansas State University. And yeah, so today I'm going to be talking about phylogeographic concordance of across the Great Plains, looking at small mammals and a lineage-specific diversification community assembly for these mammals through time. So before I get too far into it, I just want to start by going over just some quick background. So looking at what phylogeography is. So phylogeography, put shortly, just looks at how geography and historical processes have shaped diversity within a species. And so the way this is done is you take species distribution. So this here is a distribution, the full geographic distribution of a single species. You combine that with what we know about the phylogenetic data for that species. So this phylogenetic tree is showing that there are 4 distinct lineages that persist within this species. And by combining those two things, we can get the distribution of genetic lineages for a given species. And so what that usually looks like is that the lineages will kind of be regionalized throughout the distribution, how you see here. And so what this more so than anything allows us to do is identify where the genetic transition zones are from one lineage to another. And again, this allows us to infer the historical processes that shape the diversity within a given species. And so now comparative phylogeography takes that same principle and now applies it across multiple species. So this is just an example of species that are co-distributed within the boreal forest. And so each of these 4 different species has 2 genetic lineages within them. The squares represent the genetic lineage that's kind of on the western distribution of each of these species, and the circles represent the eastern lineages that are distributed across the species. And so again, the dotted lines are going to represent where the genetic transition zones are, where these 2 lineages come into contact for each of these 4 species. And in this example, we see that each of these contact zones, if we map them out, kind of cluster closely around each other in this area around Manitoba, Canada, for each of these 4 species. So what this tells us is that this community, these species, have shared some type of response to past environmental pressures, or they have some type of shared history in response to past environmental pressures. And so what that allows us to do essentially is answer the question of how a community assembled through time, and particularly, answer the question of whether species in a community even have shared history. So are the interactions that we're seeing old interactions that have always been there, or are the interactions we're seeing among modern biota more recently formed interactions? And that has large implications as for the outcomes of those interactions for a community. So for example, the species that have shared history are expected to have, less volatile interaction versus species that don't have shared history. So if you have 2 species that are somewhat closely related and they don't have shared history, for example, you would expect that they would be more likely to hybridize with one another, which can have very disruptive outcomes for the genetic diversity of both species. With that same thinking, if you have 2 species that, again, don't possess shared history, they haven't been interacting with each other through time, then we would also expect that those species are more likely to transfer parasites to one another. And with that, they would also be more likely to, transfer diseases to one another. And so an example of this is a case where we can see that a disease was transferred from a rodent to a mole. And that's significant because these 2 species are not closely related. Moles are not closely related to rodents. And so what comparative phylogeography ultimately allows us to do is understand dynamics such as these, such as the emergence of zoonotic diseases, and allows us to understand regions of potential, or regions that have a high potential for the emergence of new disruptive ecological interactions. And so with that, I'm going to get into the kind of questions that I'm kind of asking with my research, starting with, what do past distributions tell us about community assembly of present biodiversity? Also, what does comparative phylogeography tell us about the diversification within species? And with those 2 together, we're kind of also looking at how does long-term climate change influence the eco-evolutionary interactions across North America? And so my study system that I'm looking at, I'm focusing on 6 species, which are all co-distributed across the Great Plains, represented by this black outline in some way. And so each of these 6 species that I've chosen, I plan to do more, but these are just the species that I have results on thus far. And so my study system is, again, the Great Plains, which has largely been kind of unexplored from a comparative phylogeographic perspective, mainly just because the Great Plains is kind of seen as a homogeneous, flat landscape classically. However, the Great Plains is actually an extremely dynamic and complex landscape, and that's because it hasn't always looked the way it does today. So today when we picture the Great Plains, we imagine all of the prairie grasses that are distributed across it. However, around the last glacial maximum, around 23,000 years ago, all of those prairie grasses that we know today were restricted down just into the southern Great Plains at this time. And that's because during this time we saw glacial sheets covering most of North America and a climate shift that pushed biodiversity southward, hence why we see the southward distribution of prairie grasses at this time. And so this map shows where that prairie grasses would have been at the time, along with where the forest would have been during this time too, represented by the green shaded area. And so now the question is, if this is where the prairie was, then where were all the prairie species that we know today? And so to get at this question, I used distribution modeling, ecological niche modeling, of each of my 6 species during the last glacial maximum. So to go about this, I gathered my occurrence records from GBIF. I performed my distribution modeling using Maxent, and I created past projections for each of these 6 species using the conditions of the last glacial maximum around 22,000 years ago as defined by world climate. And so now getting into the results of these last glacial maximum projections, the way it's going to kind of look again, each of these species distributions is going to push southward. It's kind of the general trend is that they're going to be southern distributions. And then this white area with the blue outline represents where the glacial sheets would have been at this time. So going with the first 3 species, Rhythodonamys megalotis, Ctidus basispidus, and Rhythodonamys fulvescens, we see that each of these 3 species possess kind of a southwestern distribution, primarily on the western side of North America during this time. And then now going with the next 3 species, the Peromyscus leucopus, Sigmodon hispidus, and Cryptotus parvus, each of these 3 species we see is kind of more so on the eastern side of North America during this time around the last glacial maximum. And so if we categorize these species based on where we see them, these first 3 being on the western side, the next 3 being kind of on the eastern side, we can kind of get this visualization of the communities, a western community that would have persisted at this time with each of these 3 species, and then the vertical lines represents where they would have overlapped. And then similarly with the other species, we see that they're in this kind of eastern distribution. And again, these horizontal lines represent where each of these 3 species would have overlapped at this time. And so if we take the overlap of each of these 2 communities, these centers of origin, we see that they come into contact and overlap in this region down in the southern Great Plains area, where we would have seen the interactions among these species. So what this kind of tells us is that, one, each of these 2 communities, the species in these communities have shared history, and they do have a shared history of overlap right here in the middle of where the southern Great Plains is. And so now I'm going to get into my methods for my genetic analyses that I use for going about my comparative phylogeography. And so I've used mitochondrial DNA, Cytochrome B to be exact, as my genetic marker. And I've gathered reference data as well as collecting data from sampling on my own. And so I gathered all my reference data from GenBank. And I did constructed trees using Bayesian statistics through B software. And I performed my geographic analyses for my comparative phylogeographic assessment through GIS, and I gathered data from prior literature as well as through public databases. And so now getting into the results for my phylogeographic assessment for each of these species, the way this is going to kind of work is that you'll see down in the bottom right, you're going to have your phylogenetic tree. And so each of these symbols is going to represent a distinct genetic lineage. And all of these symbols on the map correspond to these genetic lineages. And then now we also see the modern geographic distribution of each of these species shown. And these red dotted lines are going to represent where we see a phylogenetic break occurring that is associated with the Great Plains. So starting with the first species, the Rhythodonamys megalotus, the western harvest mouse, we see that there's one genetic break occurring throughout the Great Plains in this species around northern Texas and New Mexico. For Catodispus hispidus, the hispid pocket mouse, we see that there's two genetic breaks occurring within this species throughout the Great Plains, one kind of through the New Mexico and Mexico border, and another one down around Texas. For Rhythodonamys fulvescens, the fulvous harvest mouse, we see a genetic break for this species going right through Texas, again, down in that southern Great Plains region. And then for Peromyscus leucopus, for this species, we see two genetic breaks, one kind of through Kansas and another one coming through Kansas, Oklahoma, and Texas. And then for Sigmodon hispidus, the hispid cotton rat, two genetic breaks occur for this species, one kind of on the western side of the southern Great Plains and one somewhat on the eastern side. And then for Cryptotus parvus, the North American leash shrew, for this species, again, we see a genetic break occurring down here in the southern Great Plains area of its distribution. And so if we map all of these breaks together, we get this kind of clustering of all of them. And so now the kind of question is, is this actual phylogeographic concordance that we're looking at, is this showing a signal of shared history, or is it not? Because they're not exactly clustered with one another. However, we actually might expect that this would be the result, since, again, the Great Plains is relatively homogeneous in that it doesn't have significant landscape features. However, the landscape features that we do see down here in this area, there are some present, but again, they don't act as strict barriers to connectivity. Rather, they act more as a filter. So while some species might be able to get through these barriers, some species might not be able to. So we might expect that we would see variability in the location of genetic breaks for each of these species as they have varying ecologies and they might have different responses to the same pressures. But if we compare this to our niche models for the last glacial maximum distributions, we see that the southern Great Plains might be acting as a contact zone, not only for genetic lineages, but also for biotic communities. And so this zone might be a significant suture zone for biodiversity. And to kind of bring it all around full circle, this is also the area where we saw a virus jump from a rodent to a mole. And so again, this area might be a significant region for understanding emerging ecological interactions. And so with that, my kind of answers for the questions and conclusions thus far are that one, current assemblages of species in the Great Plains reflect both eastern and western centers of origin. That's long-term overlap in the middle. And then eastern and western species both share histories of diversification across the southern Great Plains, represented by those contact zones, genetic transition zones that we see. And this all together suggests that the southern Great Plains is a significant region for emerging biotic interactions in response to climate change. And then with that, I'd just like to thank everyone that helped make this research possible, especially everyone in the Hope Lab who helped with sampling. And thank you.
I have one question for you, Tommy. Is the line for Peromyscus leucopus seems to be quite a bit north of the rest of the lines? Is that associated with its woodland habitat preference, or?
It's unclear at this time. One thing to know is that for this species, all of these samples that you see that indicate this kind of northern transition zone, these samples were just recently collected. Prior to this, actually the square that you see we weren't aware that it was actually even a lineage that persists. But yeah, I don't think I can actually answer that confidently. We don't really know what is driving all of these transition zones at the moment. We're hoping to get a better understanding by getting a better time frame for all of these genetic breaks, as well as getting a statistical contact zones mapped out. Because the ones you see now are kind of just drawn on there by eye. They're not actually, there's no statistical rigor behind them.
Okay, thanks.
Tommy, what role do you feel like, if any, population size or habitat flexibility might play in some of these patterns? I mean, Elmer's point that that Permiscus is pretty tolerable of forest habitats comes to mind, and then also like cotton rats have incredibly large population sizes. Do you feel like either of these might be shaping some of the patterns you're finding?
I think for sure the contraction of all of these populations during the last glacial maximum is playing a large part in obviously shaping the genetic breaks that we see today. However, I think it's still unclear like what is particularly driving this. So as of now, I think the best hypothesis that we have for what are driving these dynamics are temperature gradients that we see occurring across this region in the southern Great Plains that might be acting on these lineages. Again, these kind of landscape features that we see could also be driving them. But at the moment, I don't think we have enough data to give insight into the actual population sizes at the time that could be shaping these dynamics.
All right, thank you, Tommy. We have to move on to our next speaker, but thank you again. Our next speaker is going to be Kenneth Scheimer, who will talk to us about coyotes in Kenny Scheimer from Central Oklahoma will talk to us about coyotes in Southern Oklahoma. So,
can y'all see that PowerPoint?
Yes, looks good.
So, as they get, my name is Kenny Scheimer. I am a graduate student at the University of Central Oklahoma, currently working with the Noble Foundation to research the comparison of passive detection methods within coyotes at some of the ranches that they own in Southern Oklahoma. Advance, please. So I'm going to start off with giving a little bit of background of acoustics and why it is relevant in terms of collecting field data. So acoustics is fairly new when it comes to terrestrial land-based mammals, but has a long history looking at bats and aquatic mammals who exhibit forms of echolocation and very specific call patterns. But these offer a little bit of a bigger range beyond your traditional camera trap option. So this acoustic recording offers the ability to derive multiple data sets of different species across an overall soundscape, as well as to be able to observe some potentials and detections of different disturbances such as windmills, traffic, or urban sprawl, and how that can shape local behaviors between individuals of a species or overall within the soundscape of different species. From this, we're also able to database and kind of isolate out the calls so that we have a record of all the different vocalization patterns and potential of unifying or distributing, looking between different species, and then developing that database forward so that we can actually potentially identify survey specific individuals based on some species that exhibit a call pattern signature. The examples I give here is timberwolves and lions, both of which in their call pattern has a very specific end note that you can almost get individuals off of. So my study site for my research was the Noble Foundation's Oswalt Road Ranch and Coffee Ranch. These are both located in Love County, Oklahoma, just above the Texas border. The two properties are about 5.3 kilometers apart as the crow flies and are within the cross timbers and prairies ecoregions. So these sites are very much characterized by a mixture of deciduous forest with grassland, as well as grassland clearing specifically managed and grazed for whitetail, deer, and cattle. When it came to our collection method, we had 29 sites across the two ranches. We had 2 field seasons occurring in 2019 and 2020. We had 3 survey periods per field season, separated by 3 weeks, spanning from May till July. Those survey periods consisted of 4 days where there were different recordings depending on method. So when it came to the camera trap, there was a 2-minute delay to prevent same visitation repeats, but these ran 24 hours a day for the entire survey period. In comparison, acoustic monitors ran for an 1.5-hour period in the evening from 10:30 to 12 a.m. The night period, we also had a morning period, but after manually processing the acoustic data, we didn't find any coyote call patterns within that morning period, only within the evening. So we opted to only include the evening in our data set to increase detections. When it comes to processing acoustic data, there is a couple of different software options. We specifically chose Raven, which is from the Center of Bioacoustics. And from there, we were able to develop spectrograms, which are the example you see right here is actually a coyote call spectrogram. I'm going to go into a little bit more detail about that here in a minute. We used these from our recordings to be able to develop spectrograms. From spectrograms, we isolated down to a specific range. So due to prior publications, we know that coyote calls exist between that sweet spot of 0 and 2.5 kilohertz. Specifically between 500 and 1,500 kilohertz is where the most common call pattern is found. So an example here, you see right here at that 0.5 and 1, this call streak right here, those are each individual howl markers. The camera traps themselves were just processed by hand, looking specifically for coyotes, and we got those results done by that. From there, we established a binary history of both detection methods. Excuse me. And we analyzed our models in 2 different methods. So we did presence modeling, where we basically took the binary history and ran it against the collection methods as a site covariate. So 1 indicating acoustics, 0 indicating camera, because each site had 2 subsites that allowed us to have that separation of data between methods, so that we were able to run a comparison. From here, we set Psi to 1 because of our high amounts of detections, following similar practices to Cove and during one of his prior studies. And then from there, we made 3 models addressing both individual methods and combined. And out of the 3 models, we opted to use the combined data because it better shows the comparison between the 2 methods. From there, we also ran a general linear model using the same data set that we use within presence. This allows for more exact representation across different layers. So within the GLM model, we can look at individual surveys, as well as be able to look across survey versus methodology, season versus methodology, and then detection across all 3 of those variables as well, just to see a better overall ideal comparison. Example below here, we have the formula that I used within R to be able to run that statistic. So starting with the presence results data. So here we have our detection rates. They're a naive detection rate for all of them. Our overall naive detection rate was around 54%. So half the time we were receiving a coyote detection in one of the 2 methods. The camera traps only offered a 14% detection, very low in comparison to the naive, and then the acoustic detections offered around a slightly better than naive detection of 56%. This shows the start of a little bit difference between the 2 because of the behavioral patterns. When we move to the GLM, we see a lot more of an interesting flux of detections. So I'm going to talk about a couple of the very specific outliers that occur here. So within acoustic survey during season 2, survey 1, we only had a 10% detection. This was because during that period, across those 4 days, there was a large amount of thunderstorms coming through. Rain dampens sounds, but also rain deters behavior. So we believe that the reason that one was so low is because the coyotes just weren't active and the rain was dampening out most of the sound that they would have produced. Within the acoustics, we see the highest detection rate occurring during that same survey period, meaning that they might have been still walking around more or trying to find shelter, so they would have a higher chance of crossing in front of a camera. Across both, though, we can see that the acoustics generally performs better between a 40% to a 65% detection rate across most surveys, excluding the outlier of the 10% during that one survey. While the camera trap survey only shows between a 3 and 27%, with 27 being the highest between the two. So from this, we can see that acoustic does offer a better detection for this highly vocal species. They are very elusive, but they are ubiquitous across most habitat range. Thus, they're able to be picked up on both methods. But from this, we recommend that whenever it is a species that is highly vocal, to either supplement or use acoustic ARUs to be able to pick up on these sounds. And from this, other recommended research is looking into other species. So we don't currently have a database of terrestrial mammals similar of all the different vocalization patterns. We have similar ones that exist for birds and bats and different orcas and whales, that we have a full record of all the different species. We don't have that yet for terrestrial mammals, and that would be something that would be extremely beneficial, especially as acoustics moves forward. Some of my personal future research is I'm looking at AI-driven detection between the Kaleidoscope program and then the R software using the Unmarked package, where I developed basically image formattings where I can use those to run against spectrograms to detect calls. And within Kaleidoscope, doing cluster analysis, where it lumps all the sounds across the soundscape together by similarity within the range that I input for the call pattern. And then from there, we can identify which clusters coyotes occur in. And the other thing I'm going to be looking at is potential covariates that affect both detection methods. So this includes the use of climatic data, the use of distance from road, distance from water, and difference in vegetation density across both properties to see if any of those factors contribute a little bit more to occurrence and detections. I'd like to acknowledge the Nibble Foundation for their help conducting the surveys, Dr. Laverty for his help with the statistics portions of this research, Dr. Messick for serving on my board, and the UCO RCSA program for assisting with funding most of my research. Thought I had a question slide. I guess I did not. But I'll take any questions from this point.
Ken, do you have any idea how far you can detect sound, coyote sounds, for example?
That's going to be highly dependent upon which ARU you're using. So in our survey, we use a Swift monitor. According to the company that produces it, it has roughly right around 100-meter span around it. As the further you get away from that central point area, the more distorted the sound occurs. So it's kind of like with camera traps. It's highly dependent upon what model you choose and what settings you have them set up.
So do you think eventually this might be used for something like grasshopper mouse or anything else that's...
I've seen, across my reading and stuff, I've seen examples in a lot of Canidae species, a lot of Felidae, a lot of bats, but I've also seen a lot of insect ones up at the tall grass prairie up in northern Oklahoma. They have examples of specific cricket species they were looking into and looking at their behavioral breeding calls that you can extract from very similar acoustic data. It's just the monitor is much different and the frequency you're looking at shifts. But for the most part, most of the research currently with acoustics is generally based in insects, birds, bats, whales, dolphins, species that use echolocation, or species that are very, very vocal like birds.
Thank you.
Yeah, absolutely.
Thank you. I think we can pick Kenneth again. I'm going to shift to the next speaker, who is our undergraduate speaker for this session. It's Zane Sivanek from Truman State, who will talk about some effects of restoration on some small mammals, or on some mammals. Okay, so Zane, you should be able to do the share screen. Here we go.
So hello, I'm Zane Siebenek. I am a biology major at Truman State University, and today I'll be talking about the research that I did over the summer in collaboration with Dr. Stephanie Foray about savanna restoration and its effects on some mammals. So humans have substantial impacts on all of the different environments that they visit, be it through agriculture, urbanization, or the two more relevant to this discussion, cattle and the prevention of wildfires. These various factors can have impacts on these communities that can potentially alter them into an entirely different kind of community structure. Missouri, historically, was covered in about 30% of what was categorized as savanna, which typically savanna is defined with various prairie grasses and forbs, and comprising about 30% of the entire area composed in that tree coverage. But today, less than 1% of Missouri would fall under the category of savanna, and they have since been converted into forests and woodlands through these different human interactions, forests being far more covered in a canopy with shade-tolerant trees and a more thick underbrush. And various different agencies, like the Missouri Department of Conservation, are now working to try and restore these altered forests and woodlands back to their historical context into a savanna. The Union Ridge Conservation Area is the study area that I looked at in this particular study, and this area was historically largely a savanna before various cattle farmers came onto the land, and through the grazing and diminishing of the prairie grasses by these different cattle animals and the prevention of wildfires, which would burn away a lot of these tree seedlings to try and prevent any of the proliferation of these large trees. Once the Missouri Department of Conservation took up that land again, the stage was perfectly set for the area to alter and convert into a forest or woodland. And today, the Missouri Department of Conservation is now working through thinning, fire, and different restoration effects to attempt to revert this altered forest back into its historical savanna context. And so this is the region of interest. It is in northern Missouri, and everything within these red lines is considered the Union Ridge Conservation Area. More specifically, where I looked at is down here on the bottom right. The region is in different stages of restoration. This small area to the left here is the site that was performed for this study. Before 2020, this was a largely undisturbed region, but coming towards the end of 2020, thinning started to occur, and the study was set both before and after that event. This middle section is completely undisturbed, and this region to the right had been thinned and also burned. So in various different stages right now, the conservation area is. And this large center area, whiteness here, has been considered successfully restored and would now fall under the purview of what is considered a savanna. So restoration can occur in different phases, and typically does. It starts off with this thinning here, which is the simple chopping down of a few trees, which is often followed by a burn to try and burn away that understory and those different seedlings of the plants. And this process is repeated multiple times over the course of many years to eventually, hopefully, get to the restored savanna state. And this restoration process is sure to have some effects on the species within. So I wanted to look at how this first early stage of thinning was affecting the usage of the land by the different animals within the Union Ridge Conservation Area. And to this end, different sites were picked within the specific study area that we looked at. In 2020, there were about 20 sites chosen, and camera trapping was used to this end. And through different malfunctions in 2021, only 16 were used, and further malfunctions towards the data analysis portion, only 12 sites in total were actually used to be analyzed. The camera traps themselves were set up from before in 2020, before this thinning event, and after in 2021. These were set up for 2 weeks at a time, and for each of the different years, they were set up for about 168 trap nights, and a trap night being a 24-hour period in which these cameras were actually set up. The cameras are set to take 5 consecutive photos if at any point movement was detected at a 3-second delay. For the data analysis portion, all of the photos were looked at individually to try and identify any specific species. If any of the images were blurry or potentially ambiguous, they were removed from the data analysis to give the most conservative effort or conservative outlook. We wouldn't want to inflate the data saying there are more raccoons than there actually are. And so here are some of the most common species that were observed in these camera photos. And these are some of the more rare, the bobcat, cottontail, so on and so forth. And so how these photos were analyzed was on a presence and absence basis daily. So for each species, the photos were looked at, and on any given day, if they were observed, they would be given a 1 to indicate presence, if not, a 0 to indicate absence. And this was done across both years. This graph here shows the number of presence observations for each species over all sites. And the typical trend was that in 2020, there were more observations than in 2021, the biggest exception here being the white-tailed deer. And to further analyze this data, a detection frequency was found, which essentially took the number of observations divided by the number of trap nights that it ran, and then those were averaged across all sites to give what is essentially a percentage chance that you would catch or capture a presence observation on any given day. So say a raccoon had a 5% detection frequency, that would mean on any given day you'd have a 5% chance to capture a raccoon. And displayed here are the ratios of the detection frequencies, the inner pie chart being in 2020, the outer in 2021. You will notice that in 2020, it seems that these raccoons and the white-tailed deer, which are separated by these various colors, were about the same, whereas in 2021, the white-tailed deer were by and large the most captured species with the highest detection frequency. And these numbers on the outside represent the percent change in the detection frequency. So, for example, the white-tailed deer was seen 4.33% more, some more significant observations being 4.6%, 5.22%, and 1.4% for the turkey in the blue, squirrels in the yellow, and the raccoons in the blue. And through a paired t-test, the 2020 data and the 2021 data for the detection frequencies were analyzed to see if any significant observations arose. And it was found that none of these species were significantly changed in their detection frequency from 2020 to 2021. Site usage varied across the years. These are the most abundantly used sites. Site 90 here comprises some water, and that was used more often in 2021 than in 2020. Typical trend was there was a high number of observations in 2020 compared to 2021, with a very few exceptions. And you can see on this image there are some trails here across these different sites, and those are notable because some trends arose in how these sites were being used. That being that the edge sites were the sites that had the most observations, as well as near water sources. And this is likely due to animals using that trailway as easy access to traverse this different region, and then they'll perhaps branch off from the trail itself into those edge sites for foraging purposes. And being observed highly near water makes sense, water being essential for all life. It would capture quite a few animals in that region. And so there were some expected species to be seen and some more surprising results. Expectantly, deer, raccoon, turkey were all abundant across Missouri, especially in forested regions, so it's not exceptionally surprising that these were captured at a high rate. Likewise, bobcat and coyote, being top predators, would naturally have a fairly low population value, and therefore they weren't captured quite so often. Surprisingly high were things like squirrels, mice, and birds. The cameras aren't really set up to capture these species quite abundantly. They're more evasive and small to get a high capture rate. So getting these as often as I did was quite surprising. But surprisingly low were things like skunks and rabbits, which were not detected nearly as often as I thought. The camera traps seem fairly adjusted to capture these kinds of individuals, and the low presence might indicate they just are not abundant in Union Ridge itself. So it was found that none of these species were significantly changed in their detection frequency from 2020 to 2021. And that could mean that restoration isn't having any substantial impacts on these species. But I think it's important to note that this thinning event is the first step in a long process of restoration that can take up to decades. So just because this first thinning event didn't seem to impact a lot of these species, that does not mean that there couldn't perhaps be more significant events occurring in the future. It would be important to note after a burn how these species might change, and upon further review of information for this study, these small, slight changes that have been detected early on could become more significant to the point to where they become substantial. But as it stands right now, this is a solid baseline for further studies of restoration into the future. And I would like to thank the McNair staff and program who helped fund this research. I'd also like to thank Missouri Department of Conservation that also allowed use of Union Ridge. I would like to thank Dr. Stephanie Foray, who was a massive help and contributed, and we were basically partners in this whole expedition. So it was extremely helpful in getting the whole research process down. And now I am open to questions.
Any quick questions for Zane?
Zane, so I actually live about 10 miles from Union Ridge and have utilized it a lot over the years since it became an area, but that area is open to hunting. Have you thought about maybe contacting hunters and seeing if they have cameras out and if you could access their data to get a little more base there?
No, I didn't actually think about hunters possibly setting cameras out, so I'd have to get in contact with conservation. I know that a permit was needed for us to set up traps, so that probably could be a good outlet to get more data. So that's a good thought. I appreciate that.
Okay, let's thank Zane one more time, and we'll get situated for the next talk, which is Tommy Galfano from K-State, who will talk to us about some more shrews. Now we got least shrew, we got mass, now we're going to least. So take it away, Tommy.
Okay, let's see. Is my screen being shared or no?
It is not being shared, but you should be able to share it.
Okay, let's see. So I apologize. I am running into, I need to grant system access to share my screen apparently. So, oh,
Now try again.
okay. There we go. This looks better. Okay, perfect.
I don't think you were in the meeting when I did it before, so I had to do it. So anyway, you're good.
Perfect. Awesome. Thanks everybody. And hello, my name is Tommy Galfano, and today I'm going to be telling you a little bit about some of the research I'm doing for my master's thesis at Kansas State University, entitled Conservation and Taxonomic Assessment of the Leaf Shrew Cryptotus parvus. So first of all, I'd like to acknowledge that none of this work would have been possible without access to numerous indigenous lands where people were forcibly removed from, and that's for our own personal sampling as well as samples that we receive for natural history collections. So I'd just like to acknowledge that none of this work would have been possible without that. So thank you to them. And moving forward, I'm going to start talking about the genus Cryptotus. And as you can see by these images here on the right, they're a morphologically cryptic group of shrews found in the New World, and it's comprised of about 49 species. And you can see here, there's 17 species in South America, 17 species in Central America, 13 alone in Mexico, but there's only 2 recognized species within the US. So there's this hyper diversity throughout Central and South America, but only very little recognized diversity north of Mexico, which is something to note. Looking more specifically at Cryptotus parvus, this is one of the only 2 species of Cryptotus that's found within the United States, the other being Cryptotus berlandieri, which it barely creeps its way down here into southern Texas. It used to be a part of Cryptotus parvus and now is recognized as its own species, and this is its distribution right here. Oh, that meant to go back. There are 2 recognized subspecies of Cryptotus parvus within the United States, which is Cryptotus parvus floridanus, found here in Florida, and Cryptotus parvus parvus, found throughout the bulk of the central United States. This species tends to associate with mesic grassland habitats, so just have grassland habitats that have some kind of water present, but it is more tied to the presence of water in the western, more arid parts of its distribution. But recently, there have been range extensions of Cryptotus parvus discovered at the western periphery of its range, and this has happened over the course of the last 30 years, starting with New Mexico in the 80s, moving all the way up to as recent as 2006 in Wyoming. And you can see here on the map on the right, which was created in 2008 by the IUCN, that 4 of these 5 peripheral populations aren't even recognized by the most prolific conservation union in the world, which is important for conservation and management implications moving forward. Looking more specifically at populations of Cryptotus within New Mexico, there were 3 original populations that were found in the state beginning in 1985, pictured here. One at Tucumcari Lake, which is associated with the Canadian River drainage basin. One at Salt Lake in eastern New Mexico, which is associated with the Red River basin in Texas. And one at Bitter Lake in southern New Mexico, which is associated with the Pecos River drainage basin. There was recently a specimen found at the CU Boulder Museum that dates back to 1961, found there at the Red Triangle. Unfortunately, habitat in that region is completely degraded, like preventing the idea that shrews could be existing there today. And there are fossil records dating back to the last Pleistocene visualized here with the blue diamonds, which suggests that the distribution may have been much larger over different periods of time. Currently within the state, it has been identified as a species of greatest conservation need, and it is listed as threatened as well. Which brings us to the first kind of assessment. Back in the 90s, a group called Hafner and Schuster did some work on populations of leaf shrews within New Mexico. And they did a morphological and a genetic assessment. And basically, to simplify it, what they found is that Tucumcari Lake and Salt Lake here, outlined in green, fell sister to each other in both morphological and genetic assessments. Meanwhile, contrary to what you would think due to geographic proximity, this southern population found here at Bitter Lake was grouped out as an outgroup in both morphological and genetic assessments, while populations here in northern Texas at Wichita Falls fell out more sister to these northern New Mexico samples than their more geographically close Bitter Lake population, which was something very interesting to note. And so these preliminary assessments set up the background for all the work that I'm doing now. And basically, they hypothesized that morphological and these genetic assessments of these New Mexican populations indicated that populations in the north and the southern part of the states might represent distinct taxa. The specific hypotheses for these populations would be that populations in the northern part of the state represent a more recent westward expansion. Meanwhile, southern populations might represent a relic population of that species Cryptotus landii from southern Texas. And then more concisely put, moving forward, that northern and southern populations will require different criteria for future management and conservation. And so in order to address these hypotheses, we're using a combination of mitochondrial and nuclear DNA loci for both a population genetics study as well as a phylogeographic study. We're using the mitochondrial cytochrome B gene as well as nuclear SNPs acquired through dd-RAD-seq. We selected the mitochondrial cytochrome B gene because it's maternally inherited, which gives us a view of unidirectional inheritance, as well as it's quickly evolving relative to the nuclear gene. And it's a common workhorse locus that's used a lot of times for assessing levels of divergence and as well as assessing identity in groups of mammals. We use nuclear RAD-seq because which RAD-seq stands for double digest restriction site associated DNA sequencing, where essentially we take the nuclear genome and we add in 2 restriction enzymes with common cut sites, and they sample all these tiny sequences from across the genome, allowing us to get data from across the genome, but without the computational intensity of using a whole genome for our analysis. The samples that we acquired, we got about 175 total samples acquired through active field surveys and natural history collections, which are all the collections that we pulled from are listed here. They were invaluable in our project and we wouldn't have been able to do it without them. But we did active field surveys across Kansas. Pictured here, there's a shrew we caught down at the US-Mexico border in southern Texas in March 2020 as well. So that's where all of our samples came from, and they were typically either muscle or liver samples. For the mitochondrial DNA, we extracted the DNA from the muscle or liver tissue using the QIAGEN DNeasy kits, and then PCR was performed on each sample to isolate and amplify the cytochrome B gene. And then once the samples were sequenced, they were visualized, cleaned, and aligned using the Genius Prime program. For the nuclear DNA, we took the same extractions that we used for the mitochondrial DNA, but we quantified the amount of DNA in each sample, and samples with ample amounts of DNA were selected and then sent off for RAD sequencing at the University of Minnesota. And then after being sequenced, all these reads were run through this pipeline that you can see in the image here on the right, which is the STAX pipeline. And it helps clean, trim, and pare down all of the reads into what we call single nucleotide polymorphisms. And after all these filtering steps, we were left with 11,323 single nucleotide polymorphisms for our nuclear DNA data set. So now I'll walk you through some of the results. My project is a range-wide phylogeography, but today we're going to be focusing more specifically on results only pertaining to New Mexican populations. So first here, this is the mitochondrial cytochrome B phylogeny. And I know it's a lot of samples, but I'll walk you through it. It's a lot more simple to understand. We see here like a few distinct clades that group out concordant with geography. The big one, which we're going to be focusing on today, is in purple, and that's the range-wide clade. And this has samples from New Mexico, Texas, Kansas, Colorado, South Dakota, etc. But what I really want to point out in this clade is that there's no statistically supported structure within the clade that indicates that there's any internal population structure just based on mitochondrial data. All the orange bars represent samples from southern New Mexico, while the purple bars represent samples from northern New Mexico. So you can see that there's no statistical support for any differentiation between northern and southern populations solely based on mitochondria. And when you look at pairwise divergences between these groups, it's less than 1% divergent mitochondrially. Then moving forward onto the nuclear SNP phylogeny, here we see similar results with the northern New Mexico samples. One of them you'll notice is highlighted here in orange. It was also highlighted on the Site B phylogeny. This was a sample that was collected in southern New Mexico where all of these samples were collected as well, but identifies both genetically with mitochondrial and nuclear loci as northern, indicating that there is some level of connectivity between these populations. But what we see that's different on this southern New Mexico, or in this nuclear SNP phylogeny, is that the southern New Mexico group forms a statistically well-supported monophyletic clade, indicating that while mitochondrially they might not be different, there is some nuclear difference between these populations. Now, this is what's called a Pca plot, and this essentially just shows spatial differences between populations. This is using only neutrally evolving loci, and this is for all samples included in the analysis. And you can see that on this x-axis here, which explains about 10.1% of the divergence, You can see that New Mexico North population highlighted here in dark purple and New Mexico Southern populations here are quite distinct based on the all neutral loci. And now if we zoom it into both just the New Mexican populations for a little bit finer resolution, we can see that the same result is shown on this Pca plot where southern New Mexico populations and northern New Mexico populations group out incredibly differently. And you might wonder if that sample caught in southern New Mexico would be this one right here, but is actually found down here, which is indicating that it's not some example of like an F1 or F2 hybrid. This is a structure plot, and so this shows internal population structure within our samples. And so samples are organized by the capture locality, and then they're colored by their genetic identity. And so what we can see here is that the best K value or best number of internal population subclusters is 2, indicating complete distinction between northern and southern New Mexico. And we can see again that one sample that was caught in southern New Mexico that is genetically northern is entirely genetically northern and doesn't show any evidence of hybridization between these two groups. So some of the management implications of these things is that there's a large gap that exists between units that we use for management and conservation and the units scientists are able to define using genomic data. Conservation relies on units like species and subspecies, while genomic data can tell us more about like evolutionary significant units, management units, and adaptive units. Our nuclear phylogeny shows a well-supported monophyletic clade containing only southern New Mexican samples, which shows evidence that they could be considered a subspecies. But the issues with that is that they currently do not conform with any of the current subspecies distinctions within Cryptosparbust. And for us to describe a new subspecies, genomic data alone is not sufficient. We would need to incorporate morphometric analyses. So in conclusion, populations in southern New Mexico, while they're genetically distinct, they do not represent a distinct species from populations in the north. We recommend that increased sampling across these mesic habitats of the Eastern Plains is necessary, especially in the area where they caught that sample that is genetically northern. I would love to see if there's, with increased sampling, if we find more samples that maybe are hybrids or not hybrids. And then we would need to do some morphological assessments of southern New Mexican populations in order to confidently assign a subspecies distinction, but barring any kind of taxonomic revision, and making sure that further degradation of that habitat doesn't continue.
All right. Thank you, Tommy.
Yeah, thank you so much.
We're going to wrap up our morning talks now with our final talk. Kaylin Branham, who from Central Oklahoma, who will talk to us about public perceptions of mesocarnivores using a survey technique. So, Kaylin, you share screen, gold, and take it away.
Right, can you see everything okay?
Yep, you're good.
Awesome. Okay, so my name is Kaylin, and I study, I'm working on my master's degree at the University of Central Oklahoma. And this is a small section of a larger study that I'm working on eastern spotted skunks. So to begin, I'd like to move this. I'd like to bring some context to why we chose to conduct a human dimension survey. There's currently a range-wide effort to gather distribution data for eastern spotted skunks in order to gain a better understanding of the species biology and distribution. So my thesis is basically centered around using camera traps to find occurrence data within Oklahoma to add to that distribution data. The challenges with this study include a 4.5-hour trip between where I live and go to school and my study area, along with their reduced population density and their elusive behavior, which also includes them being strictly nocturnal. So the odds of me actually observing them in person are very low. In addition to eastern spotted skunks, we also detected coyotes, bobcats, gray fox, raccoons, possums, and striped skunks, and each of these species are also subject to legal harvest in the state of Oklahoma. So human dimensions is kind of a combination of different things. One is how and why humans view natural resources the way that they do, how humans want resources managed, and then how humans are affected and affect the way that natural resources are managed. So citizen science is the collaborative effort between the public and researchers to collect and analyze data. And these two points are important in this next this next part of the study, because I needed input from people who had lived there and who were in that area all of the time and had more opportunity to see these animals. I needed their insights on what they had. So the goals of this study were to contribute more efficient wildlife management decisions by providing data for this species of conservation concern, along with those other harvested species that are often understudied. It also gave us an opportunity to identify various stakeholders in the area and understand recreational land use. So the survey area included the survey area that I placed camera traps, and those included Cherokee County, Adair County, Sequoia County, LaFleur County, and McCurtain County. So the survey distribution occurred through two different ways. One, we mailed postcards with information about the project and survey instructions. We had a goal of 500 responses. So according to Betsy York, the human dimension specialist for Oklahoma, she indicated that there was about a 20% response rate for these types of surveys. So we sent out 3,000 postcards to 3,000 different random addresses throughout those 5 counties in Oklahoma. And then we also sent out an additional 1,500 follow-up postcards to addresses that didn't respond. So the survey design, we used Qualtrics to conduct it. Qualtrics is an online survey platform, and it's a really integrative platform that allows you to do a lot of different types of things. And a lot of questions are really interesting, because especially one with a map, which I'll get to later, you can actually put in places where you've seen one. So the first section is the general mesocarnivore perceptions. The first question in that section was whether or not they believe that each of these animals might be a threat to livestock, the perceived overabundance of each of those animals, whether or not they were beneficial to their habitat, and the rate of observation, whether it's decreased or increased to that respondent. And then the second section included eastern spotted skunk information, where the first question asked if they'd observed an eastern spotted skunk. If they indicated that they had, there were additional questions for them to answer, such as when they observed it, where they observed it, additional observation information that was kind of an open-ended question where they could add what they wanted, and then contact information if they'd like to talk to a researcher about their experience. The third question, or the third section, included the respondent information, such as postal code, the living situation, the time spent outdoors, species of livestock owned if they did own livestock, their outdoor activities, and then those basic demographic questions, including gender, age, ethnicity, and education. So our response success, we had that goal of 500 responses. We only actually got 123 total responses, and within that, 66 responses were with the code from postcards, which was a really low percentage of what we anticipated, and then 75 responses were actually within the study area. So of those 79 responses, with the first question, I believe that the following animal is a threat to produce or livestock. We had a number that strongly disagreed, and then those who didn't have an opinion either way, and then those who agreed, and then those who did not know. So it seemed like bobcat and coyote, which was expected, seemed to have more of a perception that they were a threat to produce and livestock. The next question, I believe the following animal is overabundant in my area. Strongly disagree, somewhat disagree, neither agree or disagree, strongly agree, somewhat agree, strongly agree, and then I don't know. So it seems like most people did not agree that they were overabundant. A lot of people seem to think that they weren't. I believe that the following animal is beneficial to the habitat it currently occupies. We had very few people who disagreed and had no opinion. Most of the people who responded actually agreed that they were beneficial to the habitat that they occupied. I've noticed the following changes in sightings of the animal. We had a notable decrease, a slight decrease, no change, a notable increase, and then I don't know, which makes sense for eastern spotted skunk since most people who were asked the question whether or not they observed one said no. So out of everyone who answered that question, only 13 said yes. And of those 13 yes answers, we had, when was the last time you observed one? 3 within the last year, 3 between last year and 5 years ago, and only 1 between 5 and 10 years ago. And then there were some that were unclear, like when I was a kid or a few years ago. Additional and observation information, we had 2 responses, only 1 was relevant. The other one was discussing how much they value mesocarnivores, which was really, which is nice to read. And then the last question, would you be interested in speaking with a researcher about your sighting? 4 out of those 13 said that they would and did give their contact information. So the next section, potential biases. How would you classify your living situation? So a majority of the people who responded lived in rural areas. 14% were in suburban areas, and then 5% were in urban areas. And of the people who indicated that they spent time outdoors, it seemed like the general trend was that most people actually spent quite a bit of time outdoors. So that was really interesting to see too. The next question, which of the following outdoor activities do you participate in? We had a lot of people kind of all over the place, honestly. It was really interesting to see the variety of outdoor activities that people participated in. What is your gender? 36% were male and 64% of the people who responded were female. And there were none that identified as other. What is your age? Most of the people who participated in this study were between the ages of 45 and 64. How would you describe yourself ethnicity-wise? So 67% recorded themselves as being white, 29% American Indian, 3% Hispanic, 0% Asian, 0% Black or African American, and then other represented 1%. What is the highest degree or level of school you've completed? Less than high school graduate, some college, 2-year degree, 4-year degree, professional degree, and then their doctorate. And the results, or the discussion, I'm so sorry. The future directions, we anticipate comparing the respondent information that we have to general perception answers to see whether or not those different biases and demographic questions have an influence on how they view these animals. We're also looking to compare the results from eastern Oklahoma to those other results. So we had some were in Washington, Texas, and then there was one in Mexico. So I'm not sure how that got there. Contributions of this study, we think that, or we made 4 connections with community members who have observed eastern spotted skunks recently. So that might actually give us an opportunity to survey areas where they actually have seen them recently. Additionally, we assessed 6 animal species that are not heavily studied due to their assumed abundance. So, and then we also kind of provided a glimpse into the societal views of mesocarnivores in eastern Oklahoma. Our survey size was not very large, so in the actual, I'm sorry, I blanked. Any actual data that we can assume is not really relevant, but it could actually provide us with means to conduct more intensive studies to achieve better survey samples in the future. So with the acknowledgements, I'd like to acknowledge Betsy York, the RSCA grants, and all survey respondents and everyone who helped distribute this survey. So with that, I will take any questions that you have.
We have a few minutes for a question or two. So have you had a chance to contact those people that said?
Not yet. I'm really excited to, though. The people who indicated that they would like to be contacted seemed really excited in their answers about it. So I'm excited to talk to them as much as they're excited to answer that question.
Were those, I might have missed it, were any of those the people that actually had seen a spotted skunk in the last 3 to 5 years?
Yes, they were. Most of them had actually seen one within the last year.
Cool.
Do you know what activity was associated most with those who had seen skunks recently?
I haven't looked at that yet, so I just processed this data very recently. But that's something that I really am interested in looking at to see if maybe there's a specific activity that might make you more likely to see them. I think that that's a really important question.
You're lucky that you're working with such an obvious species in terms of identifying it. But I mean, do you have confidence that everybody can identify spotted skunk?
Well, I'm not sure, honestly. I put pictures in the survey. I probably should have mentioned this for each section. So I had a picture of a spotted skunk that they could look at before they answer those questions. So I'm fairly confident, but some people see mountain lions when they see tabby cats, so it's sometimes hard to tell.
Yeah, no doubt. I mean, certainly some of those other mesocarnivores, the different foxes, for instance, are quite often, misperceived, I guess. But
Okay. Let's thank Kaylin real fast. That wraps up our the oral presentations. So we're going to switch now to the poster sessions. So before we start the poster sessions, just a reminder, Madison and Lori are going to give quick little spiels. I set 5 minutes, but it could be less than that if you want to do it that way. And then I'll set up 2 rooms that you can just pop in to talk to Madison or Lori about the posters, some specific questions, that sort of thing. We'll also count this kind of as a break window. So bathroom break, whatnot. After that, I'm going to leave the meeting open. So things will still, you can still pop in or whatever. Okay. We should definitely meet back at 11:30. That's when we'll do the keynote address. And at that time, we're either going to do it, I'm going to play it as a video. If that's not working well, we've got a YouTube link for the video as well. So,
hey, Sean, for those of us more challenged with using Zoom, when you set up multiple rooms, how does that, is it going to be obvious how to get in either one?
Yeah, you can join, there'll be 2 breakout rooms, and you can just join either one. Okay, So I'll try to label one as Madison, one as Lori. I think I can do that, and then assign those two to those rooms, and then you can just pop in and see the two posters.
Okay.
And I believe they should be able to share their screen within the breakout room. If not, I'll try to fix that. Okay. All right. So we have two posters. Madison Law is from Central Oklahoma, and Central Oklahoma, I think, right? Yes. Okay. She's our undergrad. Here's her little sales pitch.
Hi, can everybody see my screen?
Yes, I can see it.
So, hi, I'm Madison Law. I'm a junior undergraduate biology major at the University of Central Oklahoma, and I'm working under the advisement of Dr. Vicki Jackson on this project, where we will be observing the effect of lunar light on nocturnal rodents in a mixed grass plains region. So to start off, we know that predation risk, foraging techniques, and habitat preferences are all affected by temporal niches. And this is especially true if you're a nocturnal rodent and you're a major prey item in the ecosystem. You want to ensure that you're foraging for food at a time where your predation risk is the lowest. This means that you would benefit from being a nocturnal species that can forage under the cover of night. However, that poses the question, how dark is dark enough? So does the amount of light projected from the moon directly affect the nocturnal activities of rodents? As of now, there's little recent literature to support this notion. So we are setting camera traps as a mode of data collection for nocturnal rodents in a mixed grass plains region at the Silman Living Lab in Freedom, Oklahoma. Our objectives are to 1, measure the variation in lunar emissions in month-long intervals, 2, observe the consequential change in nocturnal rodent behavior in relation to the percent lunar emissions using camera traps, and 3, contribute useful real-time data to 2 prominent databases, Snapshot USA and UCO's Natural History Museum. Snapshot USA is a collaborative project across the US and in parts of Europe to compose a massive database of mammals in their natural habitats by way of camera traps. UCO's Natural History Museum is also working on a database for the Silman Living Lab property, and that is by Dr. Michelle Haney. And so with that, our cameras are our main source of materials. We will be using Reconyx camera traps that are programmed to take a series of 5 pictures when triggered. And our camera models are also used to measure the lunar emissions each night when deployed. So this is how we will know which moon phase inspires the most nocturnal activity, if any at all. Since we have designed the project to be fit for 2 different databases, this will be reflected in how we bait the traps. For Snapshot USA, through September through October, we will not be baiting the cameras. Snapshot USA asks that we do not bait the cameras and that we place the cameras at least 50 centimeters off of the ground and that each camera is at least 200 meters apart. From November through February, our guidelines for contributing to the UCO Natural History Museum are to bait all of the cameras with peanut butter oat mixture and set the cameras no more than 50 centimeters off of the ground. And we also are going to angle them towards the ground for optimum rodent observations. And we also ask that there will be no limit to the distance between cameras. And the moon phases will be defined as new moon with 0% lunar emissions, waxing and waning crescent in between, and then first and third quarter moons at 50% lunar emission, waxing and waning gibbous moons in between, and then finally the full moon at 100% lunar emission. Distributing the moon phases in this way will allow us for the best method of data analysis. Our data analysis will be measured by a goodness of fit test, but that is completely contingent on the nature of the data we collect in the coming months. So the fitness of data will be measured based on how close the actual data is to our predicted model, where we assume equal activity for all of the different moon phases. And since we have only collected a month's worth of data, we do not have enough evidence to draw any conclusions at this point in time. However, we have high expectations that if there is an increase in percent emission of lunar light, that there will be a decrease in nocturnal activities of rodents. Thank you.
All right, thank you. And then Lori, if you want to share your screen.
All right, can you all see that?
Yep.
Awesome. All right. Hello. So I'm an assistant professor at Fort Hays State University, and I'm going to be talking about one of our projects documenting the genetic diversity of bats in Kansas. So although there are 15 species of bats in Kansas, there have been really very few studies. I've only found one at this point, looking at the genetic diversity of bats in this state. Part of this project was inspired by some of the work coming out of Andrew Hope's lab, looking at the diversity of rodents across Kansas, and so I wanted to do the same with bats. But this is part of a larger project where we want to monitor bat population dynamics, roost use, and diet using not only tissues but also guano, so non-invasively sampled samples. Our shorter-term project, in order to have a database against which to compare our sequences that we get from guano, we need to have a reference database from bats that were collected in Kansas from specimens caught in the hand, so that we know what they were identified as initially. So to do this project, we were not nearly as far as I'd hoped. Setting up a new lab right before the pandemic started has kind of hampered our progress up to now. But we have gotten samples, wing punches that were collected by Kansas Department of Wildlife, Parks and Tourism in 2019, and we will also be getting their samples collected in 2021. They did not sample at all in 2020. I'm also using tissues from vouchers from the Sternberg Museum, as well as sequences that we download from GenBank. Because our plan is to use mini barcodes to be able to identify bat species and then insect species, we've chosen 16S mini barcodes, which are bat specific. So they only amplify bat species. And we're also using CO1. So this will give us an idea of the bat genetic diversity and allow us to amplify insect DNA from the same samples. So that will be useful when we are amplifying sequences from guano. So those CO1 primers will amplify both bats and insects. So the trees that I'm showing you here are only for CO1. I do have other information for the 16S mini barcodes if anyone is interested, and I'm only showing you a few trees at this point. So we just did traditional Sanger sequencing with GeneWiz. I used the program Genius, and we align them using Clustall. And what I'm showing you here are just neighbor joining trees, just to get an idea of the clustering. So these are not the statistically supported beast trees that we saw earlier today. These are just simple clustering trees. Very preliminary analysis at this point is that many of the bats that we have sequenced from here in Kansas have unique haplotypes compared to other species that are available on GenBank. And the bats with similar haplotypes are from pretty geographically disparate regions, and some not. So, for example, for our Laziers here, we have a Lazier sample from, let's mislabel on the poster. Awesome, you never see those typos until you actually project it, right? So anyway, we have samples from Rooks County, Kansas that are genetically identical to those from Washington State. And then another sample from Morton County, Kansas that are genetically almost identical to those collected in Texas and Saskatchewan. We also have, this is the one that is Lazieris borealis. So we have a lot of genetic diversity going on with this species, as well as the fairly widespread Nycticius. One interesting thing that we have found so far is that Yuma myotis is purported to have occurred in Kansas. We had one sample that was collected by Kansas Department of Wildlife, Parks and Tourism, and we also took one tissue or a couple of tissues from the Sternberg. And what we found is those in fact are not Yuma myotis; those are cave myotis samples. We are going to get the rest of the tissue samples of purported Yuma myotis from the Sternberg and run those. Kansas Department of Wildlife, Parks and Tourism also supposedly captured another Yuma myotis this year, and we'll be getting that tissue as well to find out if they are in fact Yuma or cave myotis. So in previous work that I've done way back in my master's, we several times actually got samples that were supposedly cave myotis that were turned out to be Yuma myotis and vice versa. So I think we might be seeing here smaller cave myotis being identified as Yuma myotis and vice versa. So what we are planning to do in my lab is we are going to sequence more samples from across Kansas. And we want to identify bat and insect species using guano samples. So we'll be looking very closely at the species of insects that the bats are eating. And we are also hoping to monitor bat roost use using guano samples as well. One problem using CO1 that we're running into is there are not nearly as many samples of CO1 on, say, GenBank compared to more common genes like Cytochrome B. But we were having problems getting full Cytochrome B amplified. And also Cytochrome B makes for not very great mini barcodes. So that's why we opted for CO1. The CO1 samples that are on GenBank often do not have their geographic locations associated with them. So that makes trying to figure out if there's interesting genetic breaks a little bit harder to detect. Yeah, thank you. I'm happy to answer any questions in the breakout rooms.
Okay, thank you both. I'm going to open up new breakout rooms. I'll label one as Lori, one as Madison, and you can pop in and ask questions there. You can take those rooms will be open till 11:30, and then we'll do the keynote address. So let me stop. Oh, and then the other thing is I've recorded the whole thing. I forgot to get the first couple slides from Nathan's, but I can break that up if people want their own individual talks and videos and whatnot, or need to share it with other people. Okay. Breakout rooms, let participants do